first episode was talking about the hematologist, oncologist himself. The second episode is talking about cancer and hematology. So the last time we were talking about cancer, and guys, please go ahead and open the Showcase app and click on the hematology, the cancer and hematology link to follow the presentation. Okay. So what I was saying that the last time we talked a little bit about cancer and hematology, we didn't actually get the chance to talk about hematology, but we were talking about cancer. We talked about what's cancer and the genetic basis of cancer and the cancer treatment. I'm gonna go through this stuff again with a little bit more of details, just trying to make us more aware of the genetics and stuff about cancer, because this is kind of very important thing to give you the situational awareness. The situational awareness, always Dr. Nasser and Ahmed keeps talking about the situational awareness, to think on your feet and react to the questions or the information the doctor is telling you. So I'm gonna go through them again, add some more details, and if we have more time, we can go through the hematology part. Okay? So let's start. Now, okay. So the last time we spoke about what's cancer, and we were saying that cancer is a disease that's caused by a group of causes, a gang of causes, that drives the cell to be abnormal, to have an abnormal behavior and start an aggressive behavior towards the others, and encroach on their privacy, or encroach on the other tissues and start being aggressive to them. We said that cancer is responsible for 15% of all human deaths in the last couple of years, which is pretty huge. So it's an issue, and it costs the United States a lot. Like I remember reading a report that said maybe it's like almost $1 trillion, or I'm not sure actually, but it was a huge amount of money that's spent in the cancer treatment, and cancer research, and taking care of the cancer patients and stuff like that. And then we spoke about the cancer genetics, and we were saying that the cancer is a problem of growth. And just to understand more about the kind of problem of growth, so what's happening in my body and in your body, that the cell, so we all started from the fertilized ovum, which is one cell, a 46 chromosome cell, and this cell starts to replicate, given two more cells, and each cell starts to replicate and divide, replicate and divide. And as soon as we reach puberty, most of the cells of the body, either they are in the resting phase, which means they are no longer dividing. They still have the potential to divide, but they are no longer dividing. And on average, the normal human cell has the ability to replicate only around 60 times per life, and then it's just kill itself. So this number 60 times is a really important thing, because when we're going to be talking about the 6 hallmarks of cancer, and saying that one of the techniques that the cancerous cell use to be cancerous is it escapes death and it becomes immortal. And by immortal, we mean that it has a limitless capacity to replicate. So this is exactly what happens. A normal cell would only have the ability to go through 60 cycles, but the cancer cell can go limitless. So basically, it's a problem of growth. It happens while the cells or the tissues are grown. Usually, after 60 times of replication and dividing, the cell should kill itself, but the cancer cells never kill themselves. And then we talked about the multiple genes, and here I want to stress on a point. what does the doctor need from us, the hematologist oncologist? What what? Go ahead.
You keep disappearing, at least that's what I hear. Can you repeat the last couple of sentences, please? So after 60 times of replication, replication is like normal cell.
After 60 cycles of replication, a
Mm-hmm.
normal human cell should kill itself.
Mm-hmm.
It can't replicate anymore. It just so each of our cells, it has a programmed cell death. It's kind of, a program that's in the cell itself. When it turns on, it kills the cells, like it shuts the cell down. This is kind of nature thing. It's very important to keep us healthy, because after 60 times of replication, there are a lot of alterations that are summed up. Those alterations may result in, a problematic cell, a mad cell. That's why after 60 times of replication, this cell is, old enough. Maybe it's malfunctioning. It should kill itself. This happens normally in my body and in your body and in every other person who's normal. But the cancerous cells are no longer like this. They can somehow escape this apoptosis process, which is the apoptosis means programmed cell death. So they can escape this process and keep continuing replicating forever. Okay. Then we were talking about what exactly do the doctor need from us. So he needs 4 pieces of information. Does this patient have cancer or not? To say that the patient has cancer, and then why this cancer is happening, and then at which stage this cancer is, and finally to which treatment this cancer responds. So those are uh, uh, four important elements of the diagnostic service. The 4 main pieces of information that the doctor would want to get from us. Does this patient has cancer or not? And what are the causes for this cancer? How far has this cancer gone? And then which treatment should this cancer respond? So to diagnose that the patient has cancer or not, some ways to say that this cell has turned cancerous, and this mostly when the pathologist looks under the microscope and sees the cells and their arrangement and the tissues and their arrangement and the changes that happens in them. Or we can do flow, or we can do immunohistochemistry, which tells us exactly if those cells have changed their appearance or the molecules that are on the surface. So we do flow and immunohistochemistry to say, yeah, there is cancer. But then we have to know what are the causes for this cancer. And to know more about this, we have to understand that cancer is caused by multiple genes. It's a lot of genes that are involved in the process of developing cancer. And remember the last time we had this analogy when we said cancer is like failure. So any problem that can happen in any gene that can lead to a cancerous, to abnormal cell, this is defined as cancer. We used the analogy of a broken car. It may happen because of the motor or the electricity or the body of the car. Any problem that has happened that leads to the car being broken and not able to go on the road, this means cancer. So I just want you get the idea of multiple. So to answer the second question, what are the causes for this cancer, we have to investigate what are those genes that were changed and led to cancer. And then to answer the third question, which is how far has this cancer gone, we want to understand that the cancer is kind of transformation process. So it starts as this is a very benign normal cell, and this is a very aggressive, bad, maybe even self-destructing cell. And there are different stages in between, and a hematologist oncologist would probably need to know at which level we are, how mad those cells are, so that he can decide which drug works against them best. So this is kind of trying to answer the third question, which is how far has this cancer gone. We want to understand a concept that cancer is a transformation of cell or transformation of tissue that has different stages. And then this whole process leads to the undesirable survival of the fittest. Because, yeah, the cancer cell is a problematic cell. It has a lot of problems, but it's more able to divide. So it kind of gets all the nutrients from the other tissues and pressurize them and attack them or invade them. And it's, at the end of the day, it's the fittest one to survive. So this is kind of basic background about cancer. And then we went through the idea of what exactly the genetic materials are. Just a second, I want to check. So we were saying that the normal human cell has a nucleus, and then this nucleus is composed of 46 chromosomes, 23 pairs of chromosomes, and each chromosome is a very, very, very long coiled double-stranded thread of DNA, and this double-stranded thread of DNA is composed of nucleotide bases. And here we want to, go furthermore and answer a question: how exactly the information is stored in our body? The genetic information is stored in our body. I think, Katrina, you have a good background about this, right?
Sorry, yeah, I have some.
Yeah. Can you go ahead and tell us about how the information is really stored in our body?
You mean the genetic information?
Yeah.
Oh, Karim, I wouldn't be lectured on that. I don't think I can just, kind of lecture on that.
No, I mean, just share your ideas. I mean, how this information is really stored in our body. So the idea is, the binary system, right? The 0-1 system that codes for the digital information.
Mm-hmm.
We have, instead of being binary, which means 2-element system, we have a 4-element system. They are the 4 types of nucleotide bases. They refer to them by the letters A, T, G, and C. And the information is really stored in the arrangement of those nucleotide bases. So let's say this is A, C, G, A, G, C. So this sequence codes for the information. And it can be translated to amino acid arrangement in the formation of the protein molecule. So this is the idea, is the arrangement of this nucleotide bases codes for the information of our body. And then these nucleotide bases are in pairs. So if this is A, this is T. If this is C, this is G on the other side. So they exactly go with each other. They would call it the sequence and the complementary sequence, which is the other sequence that goes with this, matches the sequence. So those double strands are matching each other, and this is kind of important information to understand how the mutation is going to happen. So basically, this is a cell, this is a nucleus, and this nucleus is formed of 46 chromosomes, 23 pairs of chromosomes, and each chromosome is formed of a very, very, very long thread of double-stranded DNA, and this double-stranded DNA, they call it DNA helix. It's composed of nucleotide bases that's arranged in a certain way that codes for the information. And what happens exactly in our lab, so the services that we provide is the hematopathologist looking at the biopsy, he can see the entire tissue, the all cells together, and can see the one cell and how changed it is. And then the flow cytometry and the immunohistochemistry sees what's on the surface of the cell. the receptors that are, or the molecules that are on the surface of the cell. And then the cytogenetics looks at the nucleus. It can see the chromosomes, but the entire chromosome. It has kind of an eagle eye view that sees a very big picture, but it can't see the details. So the karyotyping can see the entire chromosome. The fish still sees the entire chromosome, but it can see a single spot in the chromosome by light. So FISH stands for fluorescence in situ hybridization, which means that we can find a spot on the chromosome and put the fluorescence on it, hybridize the fluorescence in it. So when we look under the microscope, we can see this spot as light. So cytogenetics uses the microscope. It can see the entire chromosome, either a dark chromosome, which is karyotyping, or some lightening spots on the chromosome, which is FISH. And then the molecular see on this level, the DNA level. It can see the sequence of the base pairs of the double-stranded DNA, which basically codes for the genes. So do you guys follow this? That the pathologist, the biopsy, the Dr. Nasser looking under the microscope, he sees the entire tissue, the adjacent cells together, and then the flow and immunohistochemistry looks at the surface of the cell. Then the cytogenetics looks at the nucleus at the chromosome level. It can either see it by karyotyping, which is kind of black chromosome or dark chromosome, or it can see some spots on it by adding light to it, which is FISH. And then the molecular can see the exact sequence of the chromosome, of the gene in the chromosome. So do you guys get this idea? If you have any problem, just let me know. Have any question? Any thoughts?
Yeah, can you repeat about cytogenetics? So cytogenetics is karyotyping, and what they see, they compare the pairs, and they compare the number. I mean, they count the number of chromosomes, right? Pairs.
Yeah. They compare the numbers of the chromosomes and the shape of the chromosomes. So let's say this is how the chromosome looks like. Let's say if this chromosome is cut from here, then it doesn't look normal. The physical appearance of the chromosome, it doesn't look normal, right?
Yeah. I mean, Karim, we see it in the last page of our report. It's fish and karyotyping, and the karyotyping picture shows the all like 23 pairs of chromosomes and how they differ.
Sure. And exactly on the last page of our report, I'm going to go through this on the next slide, so don't worry. I'm going to tell you something interesting about this. But do you get that karyotyping can look at the chromosome and see how many chromosomes there are, if they just look normal or not? It just looks at the 46 chromosomes completely at the same scene, you know? So it sees a bigger picture, but with very, very limited details. Fish can find small spots on the chromosome by sequence, by the DNA sequence, but it can't go further and tell us the details. It just adds some lightning spot on the chromosome. So when we look under the microscope, this is exactly what happens. You look under the microscope, you see the 46 chromosomes. They are all dark, but you have one chromosome that has one lightning spot. Don't worry about this. The entire second part of the training is gonna be talking, the entire 6 episodes talking about what exactly happens in PsyParadigm and each department and what do they do and what are their techniques and all this stuff. So don't worry about this. I just want you give the general idea that the pathologist looks at the tissue, and then the flow and immunohistochemistry looks at the surface of the cell. The cytogenetics looks at the nucleus, and then the molecular looks at the sequence of DNA. Okay? Okay. So now when we're talking about cancer, it's all the DNA fault. So what I mean by this is, no, back. Yeah. So cancer, it's all the DNA's fault. there are three levels of the problem that can happen. Either it happens on the very, very, very big level, the chromosomal abnormality level, which means there is a missing chromosome or an additional chromosome. So of course, all of us know the Down syndrome, right? So the Down syndrome is basically a patient that has the chromosome pair number 21, and instead of having only two chromosomes, a pair of chromosomes, it has three chromosomes. So they would call it the 21st pair trisomy. And this is exactly what's in the last page of our report. When we show the doctor, we show him the picture of karyotyping of a patient has one extra chromosome. So this is a huge change of the genetic material. There is an entire chromosome coding for maybe a couple thousand of genes, different functionality. That's why those patients looks abnormal. They looks different. They have a shorter lifespan. They may have cardiac problems. They may have cancer. You know what I mean? This is a huge change of the genetic material. So the first DNA problem can happen at this very big level, or it can happen on a large scale gene mutation. So it's either chromosomal abnormality, which means a huge change, one extra chromosome, one missing chromosome, it's called Turner syndrome. When there is a missing chromosome, it's called Turner syndrome. Or a break of the chromosome, like you miss one part of the chromosome. This is a huge chromosomal abnormality. Or it can happen as a large scale gene mutations, like a small part of the chromosome gets deattached and reattached to another chromosome, and the part of this chromosome comes back here. It's called translocation or inversion. Like a small part of the chromosome gets deattached and then it turns upside down and gets reattached again. So what they would call this a large scale mutation, which means there is no extra gene or there is no loss of a gene, but the arrangement is different. So this is a large scale gene mutation. Or a small scale gene mutation, which most of the time they would call it single point gene mutation. Remember when we were talking about the nucleotide bases and the genetic information is stored in their arrangement? So let's say if there is a missing piece, a missing base pair, or a changed base pair. So let's say if this is, instead of being AT, it has become GC. So now this three adjacent base pairs is coding for a different amino acid, and this different amino acid is gonna change the entire shape of the protein coded by this gene, and this protein is gonna function in a different way. So let's say there is a sodium channel. Basically, the cell has pores. These pores allow the sodium and potassium to go in and out, and for the cell to function normally. Let's say this protein that acts as a pore, a sodium channel, is changed in a way that makes it block. So now there is no influx of sodium or outflux of potassium. This cell is not going to function normally. You know what I mean? Sometimes the gene codes for a protein that's involved in the growth cycle, or it's involved in the replication process. If this gene is coding for a protein that's involved in the replication process, any change in the base pairs of this gene is going to change the amino acid, going to change the entire protein shape. It's going to make it act in a different way. Maybe this protein was responsible for receiving the message that asks the cell to go ahead and divide. Now, instead of this protein waiting for the message, he's self-firing, and now he's asking the cell all the time to go ahead and replicate without receiving a message from the body telling this cell to replicate. So now this cell is gonna act in a different way that the body doesn't want it to act this way. You know what I mean? You get the idea? Just think of it as an army or a nuclear submarine instead of waiting for the orders from the headquarters to fire, it just fires by itself. Now we have a huge problem, or we can even make a movie about this. You know what I mean? It's a very, very, very problematic issue. So again, let's say it's all the DNA's fault. It can happen at 3 levels. Either the chromosomal abnormality, a very big level abnormality, missing chromosome, an extra chromosome, or it can happen at a large scale gene mutation, small part of the chromosome, maybe 10 genes, 11 genes, 100 genes, I'm not sure. It can be in different ways. It can, gets reattached in a different way, gets reattached to a different chromosome. So this is a larger scale gene mutation, or it can be a single point gene mutation, which means a change in one or two of the base pairs of the gene. Just to understand more and make this kind of information more, in an application way, this is exactly what happens. Of course, all of us has received a question or more about the BCR-ABL gene and the BCR-ABL test, and what's the turnaround time for us for the BCR-ABL test. So to understand this, this is called the Philadelphia chromosome, and this is a kind of a hallmark for the CLL leukemia, chronic lymphocytic leukemia. So this is the chromosome number 9, and this is the chromosome number 22. A part of the chromosome number 9 gets deattached and reattached to the chromosome number 22, and vice versa. So now on this part, there is a gene that we call ABL. A gene, just remember, a gene is a certain sequence of the base pairs, a part of the DNA. So this gene is called ABL, and this gene is called BCR. Normally, in my body and your body, those genes are very different from, very far away from each other. But now, with this translocation happening, those genes are brought together, right? And this is called the BCR-ABL gene. And this is a hallmark for CLL. So for a doctor to diagnose a patient having CLL, this patient has to have Philadelphia chromosome. So now, if he has a patient suspecting that this patient has CLL, chronic lymphocytic leukemia, now he wants to know if this patient has the BCR-ABL gene or not. So he sends to CyberDime saying, guys, please, can you tell me if this patient has a BCR-ABL gene or not, right? Then what should we do? What test, what department should handle this case? Go ahead, guys, and tell me, what do you think? Is it flow? Is it immunohistochemistry? Is it cytogenetics? Is it molecular? What are the department that can find this?
Molecular.
Molecular? Sure, molecular can find this. How? Basically, I'm sorry? Go ahead. What did you say? Hello? Katrina, you were talking?
Yes, cytogenetics?
Cytogenetics. You said molecular, right?
No, somebody else.
Okay, sure. So how cytogenetics can find this? By karyotyping or by FISH?
Karyotyping.
Karyotyping. How come karyotyping is going to be able to find this? Basically, when you look under the microscope, you wouldn't have colors, so it exactly looks like the same. This one looks like this one, and this one looks like this one.
Okay, what about light? If we use FISH, do we see it? No. Spots? No.
Yeah, we can use FISH. So what would they do is they would find a light that can, put a light on the ABL part of the chromosome. Let's say a yellow light. That's exactly what happens. And they put a red fluorescent light on the BCR chromosome. And now when they look under the microscope, they should find the yellow light far away from the red light, right?
Mm-hmm.
But if the patient has Philadelphia chromosome, they would find the two lights just next to each other, right? So now they can say that this patient has Philadelphia chromosome. Or we can do this by molecular, because we know the sequence of the ABL gene and we know the sequence of the BCR gene. Let's say, just for example, the ABL gene is composed of 30 base pairs, and the BCR gene is composed of 20 base pairs. So adding them together, they are 50 base pairs. We can do the complementary strand of those 50 base pairs and send it in the specimen, and this strand is going to find the complementary strand of those 50 base pairs coming after each other in this arrangement. And if those 50 are just next to each other, this complementary strand can attach to it and tell us, yes, he has the BCR-able sequence, right? Because in a normal patient, the complementary 50 base pair strand cannot join to either this or this alone. They have to be combined together to be able to attach to them, right? So we can find the answer by molecular.
So how would y'all decide whether to do molecular or cytogenetics through FISH?
Actually, we do both of them, just to make sure.
Just to confirm, okay.
Yeah, but then you have the next question from the hematologist oncologist. What's your turnaround time for the BCR-able test? So, Lauren, what would you say?
Well, with molecular, in my case, it takes 4 or 5 days.
Mm-hmm.
But at the same time, FISH only takes a day or two.
Mm-hmm.
So it kind of depends.
Yeah.
I guess total it would be, you would go ahead and say the 4 to 5 days, right?
Yeah. So if you get the question, you would most probably say in 24 hours. So if a doctor says, what's your turnaround time for PCR-able test? I would say, well, 24 hours to get you the results by FISH, and just 2 more days to get you the results by molecular. So I rearrange my answer to tell him, yeah, I can provide you the BCR-able test results within 24 hours by FISH, and then by molecular. And you know the difference between them is, by FISH, I can tell that there are some cells that has the Philadelphia chromosomes. So it's qualitative, but I can't know how many of the cells has this abnormality. So it's not quantitative. But by molecular, I can know exactly how many cells in the specimen consumed the complementary sequence. That's why it's qualitative and quantitative, both of them. So for a doctor who wants to diagnose his patient for the first time, he only wants to know if the patient has Philadelphia chromosome or not. So it's kind of qualitative. So the FISH results should be enough for him. But if he actually knows that the patient has CLL, the patient is doing follow-up, so the FISH results are not of very great importance for him, because he actually knows that the patient has CLL. So the qualitative results are not that important. But the quantitative results are important because they are going to tell him how many cells still have this abnormality, which means how does this patient respond to this treatment, if he's doing very well or doing very bad. So for a follow-up, he wants the results by molecular, but for the first diagnosis, most probably the results by FISH are enough. Now you started to get the feeling of the difference between them?
Yes, that was great.
Mm-hmm. Sure. So that's basically how the PCR-able test is kind of done. So the next time you're asked the same question, what's your turnaround time for PCR-able? You would say, yeah, we can provide you the results within 24 hours by FISH. And then 2 more days, we can provide you the results by molecular. Well, you don't have to go through to go further and explain to him that this is going to be sufficient if this is a first-time patient, or this is more important if this is a follow-up patient. But you have to get the information in your mind. So if he asks you, no, I'm not interested in the fish, I ask by molecular, then you would know that that he's thinking of the follow-up patients. So you say, yeah, I understand. You need the molecular for the follow-up patients, so we can provide this within 3 days of receiving the specimen. Now you are kind of more able to respond to the questions he asks, and in your mind you know why he asks this question, right?
Mm-hmm.
Okay, sure. So let's go. So this is basically how the chromosomal abnormalities work. So all our body functions are mediated by proteins. All our proteins synthesis is coded in our DNA. A bad DNA means a bad protein and a crazy cell. Now remember we were talking last time about the cell cycle. So each cell in our body is in the resting phase. It receives some growth signals that activates this cell and asks her to go through the dividing process. And in the same time, it receives anti-growth signals that asks the cell to stay or remain in the resting phase. So this is kind of very common phenomena in our body, that everything is kept by balance. So there is always a balance between signals that asks the cells or the tissues or anything in the body to do something, and signals that asks it to stop doing this thing. This kind of fine balance that regulates our body. So if this cell now needs to be dividing, it is going to receive more of the growth signals and less of the anti-growth signals. And vice versa, if we need this cell not to be dividing, so it's going to receive more anti-growth signals than growth signals. I know this is kind of confusing, but if this concept is clear, then we're going to be fine for the next part. If you still have some confusion about this concept, just let me know. So I'd assume we're fine. So for the anti-growth signals that asks the cell to remain in the resting phase, those signals, should have proteins that regulate the stuff. These proteins are coded by genes, and because those genes code for proteins that are involved in the anti-growth signals, we call them tumor suppressor genes. And the genes that code for proteins that are involved in the growth signals, we would call them oncogenes. And don't get the bad feeling that those genes are in our body just to develop cancer. No, those are good genes doing very good stuff. Just think about it. It's like an army. The army of a country it's a good thing to be there to defend the country. But if this army has gone mad and it started to attack its own people, now it's a bad thing. And we would call them, coup or something. This is just the idea. Those genes are really good genes. We need them to keep, growing and doing the good stuff. But they are kind of involved in, if they gone mad, they are involved in the oncogenesis, which is tumor developing process. And those are the genes that should that's called tumor suppressor genes. They are doing other stuff in our body, but because of their part of the tumor process, they call tumor suppressor genes. So now we have the balance between those two genes, right? Anything that leads to inappropriate activation of the oncogenes is going to ask the cells to go ahead and divide. Because this is inappropriate activation, so this division is going to be inappropriate division, which means cancer, a problem of growth. Or any problem or mutation that leads to those genes not working, this means those cells are going to go ahead and divide, and deactivation of those genes is going to lead to cancer. This is basically what this in this chart. Gene mutations that lead to activation of oncogenes or deactivation of tumor suppressor genes are going to lead to cancer development. Is this clear or still confusing? I know from the last, we said this exactly the last time, but it was still confusing for some people, right? For me, it was still confusing even. So basically, the gene mutations, any activation for oncogenes or inappropriate deactivation for tumor suppressor genes would lead to cancer. And by inappropriate, I mean either it's in the wrong time or not in the wrong, or I mean in the wrong quantity, like huge amount of activation of oncogenes. So either inappropriate time or inappropriate tissue or inappropriate quantity, this is called inappropriate. That's gonna most probably lead to cancer. Are you guys following the presentation or not? Because I see only one person with me on the presentation.
Oh, I'm looking at the screen that the main screen. I don't have it clicked on the I should be on showcase, is that right?
Yeah, sure.
Oh, okay. I was just looking at the regular screen.
Okay. I mean, you're fine if you're looking at the main screen and you see it moving, but it's gonna look more nice and more smooth when you look at the link in the showcase. But you're fine. Okay, you guys are typing stuff in the chat. I didn't know that. I'm sorry. Okay, so let's go ahead and talk about the hallmark theory of cancer. So this is this theory, when it came up in the year 2000, it was kind of revolutionary look at the cancer. It described to us how cancer works, why those cells, what are the changes that happens to those cells that leads to cancer. It's kind of like, you know, the Bible for cancer, or the Holy Book of Cancer. It just told us how the cancer works. So it's 6 steps. I tried to simplify and rearrange them in 3 phases: steady, ready, go. You know, just tried to escape the fancy words of, you know, the theory in a very kind of simple words. So basically, the first 2 steps is to be insensitive to the anti-growth signals, which means escape death. The anti-growth signals that ask the cell to no. I'm gonna start over. So the first thing is they don't have the apoptosis process working. You remember when we were talking about the apoptosis, which is the programmed cell death? So they have this program not working, so they are able to escape death. And remember when we were saying that each cell has the capability to reply to you know, for replication and division only for average of 60 times before it kills itself. Now those cancer cells doesn't have this limit. They are capable of limitless division. So basically they are immortal. So they are able to escape death and being immortal. Now they are steady. Then the next phase, they are okay. So the next phase, they are they disrupt the balance between the growth signals and the anti-growth signals. And by this I mean they never ever respond to the anti-growth signals. They never stop dividing. And on the other hand, they have self-firing of growth signals. They don't need growth signals to grow. They just by themselves have sufficiency in growth signals. Just kind of, you know, having high self-esteem. They don't need any other body to tell them to divide, or, you know, that they are good or they are fat. They just go ahead and divide by themselves. This is the second phase. And the third phase, they are able to perform blood vessels for themselves. So exactly to understand this, we have to look at the normal body. For a normal body, like let's say when you have a muscle to grow, this muscle, like the muscle cells, are able to regulate their own growth, but they are not able to provide the entire, blood vessels that needs to grow bigger, to develop more supplies. They are not able to do this, and the regulation of this is kind more of the body responsibility, like the control of the body that regulates. If the muscle is going to grow big, I'm going to provide them with more blood vessels. But for cancer, because cancer is not following the body regulations, they are just doing this by themselves. So they were able by somehow to develop their own blood vessels. So now they can provide more nutrients for themselves, and they don't have to wait for the body to provide them with blood vessels. Because just think about it: if those cells weren't able to develop their own blood vessels, they would have died. They are replicating very rapidly in a very aggressive way, but they do not have the supplies to divide, so they're gonna die. They're gonna kill themselves. But now somehow they are able to provide them, to develop their own blood vessels, bring more and more nutrients for themselves, and that's just makes them more eager and able to invade the next tissues. And they now can invade the adjacent tissues. They would be called locally malignant. They would, let's say a breast tumor would invade the chest cage, the thoracic cage, the bones under it, or it can split and go in the blood vessels to another spot and start developing a colony there. It's called metastasis. So it's just, you think about it, the tank's force, or the Navy. The Navy can go far away and start and deploy, another colony or another base overseas, or just being locally malignant and attack Canada or Mexico or whatever. This is just to get the idea. So this is called the 6 hallmarks of cancer. Just wanted you guys to get the idea how cancer works and how this affects the body. Then I'm gonna go very rapidly about the cancer treatment. We discussed this in detail last time. So basically, the first line of treatment for cancer is surgery, which means if there's a tumor in the breast, let's do mastectomy. But this can't be done for every patient and can't be done for every tumor. So if there's brain tumor, I am not able to go ahead and take the tumor off. Maybe some small tumors in some superficial places, but not any other. I can't just get your tumor out or your heart out. So it's kind of more limited to specific tumors by specific stages. Or let's say if a patient has a breast tumor, but then it has a secondary metastasis in the leg of the bone. Now we are not able to do mastectomy up there and then remove the leg and then remove the liver and then remove the spleen. You would better to let the patient die in peace, not in pieces. If you know what I mean. So surgery kind of limited. They are the most important one. They are the first line of treatment, but still they are limited to certain types of tumors, certain stages of tumors, certain patients. And then we started to develop the chemotherapy. Chemotherapy it's fantastic line of treatment, but it has a very bad problem. It's non-specific. It kind of shuts the process of dividing in every cell in the body. So it just tells all the cells in the body there is no division, no replication. The tumor cells would respect this. They're not going to divide, which means we kind of limited this cancer. But again, all other cells in the body are not going to be responsive. Think about it. If we give chemotherapy by this concept to a kid, he's going to remain kid forever. He's not going to grow up. And that's why there's a problem that happens for a patient who has cancer. That's why patients who get chemotherapy, like they start to lose their hair, because the hair follicles needs to be continuously dividing and replicating. But now, since no cell in the body is allowed to go ahead and divide, those patients start to lose their hairs or start having all those kind of complications that come with chemotherapy. That's why we started to develop the next line of treatment, which is radiotherapy. So the radiotherapy idea is the radiation destroys the DNA of the cells. But now the normal cells, they kind of have, like the cells in my body and your body, they have kind a process of fixing the DNA. So they can fix their own DNA. You know, like they have checkpoints, and they can, because you remember when we said they are double strands. So if we lose one strand or part of one strand, we can always find the information on the other strand, because each A on one strand is going to have T on the other strand. You know what I mean? They can fix themselves. But the cancerous cells can't. They lose the ability to fix their DNA, because if they have the ability to fix their DNA, they wouldn't have been cancerous cells. Because if there is a mutation and they can fix it, they wouldn't have been cancerous cells. So they are cancerous cells because they don't have the ability to fix their DNA. So now we give the patient the radiation, it's going to affect all the DNA of all the cells. But then we're going to stop. The normal cells are going to be able to fix their damaged DNA, but the abnormal cells wouldn't, so they most probably gonna die, or at least they are not going to be able to divide again. So this is the basic idea, but this is a very, very, very hard goal to achieve. We are not always able to give the right dose, the right way, to the right patient, and it's multifactorial, very, very hard. That's why there is a certain specialty, a doctor who spends 14 years just to know which dose, which way. And we can, you know, like give the patient extra beam radiation, or brachytherapy, which means we give the patient a brachytherapy, which means we give the patient the radiation from inside, like a pill. You know, he gets the to swallow it, and it just radiates from inside. It's a very, very tricky line of treatment. It's helpful. That's why most of the hematology oncology clinics we go through, they have a radiation clinic. They may have a radiation oncologist, but still we need more specific lines of treatment, and that's why we have the targeted therapy. The targeted therapy is a really important line of treatment. And remember when we were talking about the 4 pieces of information that the doctor needs from us as a diagnostic service? This is the 4th piece of information. What kind of treatment this patient is responsive to. So we need to scan his genes and know what genes are changed. And those genes, because those genes are coding for proteins, we can find those altered proteins and just target them. This is a revolutionary thought, a very, very interesting line of treatment. We can find the cell that exactly has the problem and then target it by a drug. But this is really hard to achieve because we are not able, with the current ways of molecular diagnosis, we are not able to scan for genes. Because remember when I was saying that we have to know which sequence we are looking for, we can't scan for the entire sequence of the DNA. This is a genome project. It needs years and hundreds of millions of dollars to do this job. But now we have the next generation sequencing. And next generation sequencing is a revolutionary technique that's going to change how this works. And we are finally able to scan for genes so that we can know which genes are involved. Remember when we said it's multiple genes? We would know which genes are involved and which drug just to target those mutant cells. Just to give you a number to compare, the current method of the molecular diagnostics which we are using, it's called the RT-PCR, the reverse transcriptase polymerase chain reaction, and this can only find sequences as long as 96 base pairs. But the next generation sequencing, we can look at sequences that's couple of hundreds of thousands of sequences. maybe 200,000 base pairs or 300,000 base pairs from 96, from 100 to 200,000 or 300,000. This is a huge improvement. It's going to enable us to scan for genes, know exactly what are the genes that's changed in the cells, and what exactly the mutant proteins, the dysfunctional proteins that led to the problem, and then we can target those cells with those fingerprints. Now we're going to spare all the other normal cells in the body and just target the problematic cells. For now, I don't have that much time to go through the next generation sequencing in detail, or at least give you a kind of hint about how interesting this is. Maybe the next times, or when we go to the second part and talk about the molecular diagnostics. But just I want you guys get the idea that this is the future of the industry. Next generation sequencing is the future of the industry. And now we have a lot of potential to go out there, start selling those tests, and get the doctors to know more about their patients, help more patients, and actually basically make more money. So so it's the future. Now in PsyParadigm we are still in the process of developing this new technique. Other competitors, New Genomics as an example. So in PsyParadigm now we have 20 tests. We can scan for 20 genes, but New Genomics, they develop 80 tests per year. So they are growing very fast. That's why we now hired, in PsyParadigm, we hired a doctor that's kind of specialized in the validation of the next generation sequencing tests, just to keep up with this, because this is the future of the industry. Basically, the immunotherapy, we talked about the immunotherapy last time, and you guys remember talking about the Solaris drug, which is the most expensive drug in the world, which costs almost $450,000 per vial. It's basically we get we get the antibodies that works specifically against the tumor cells and can attack them and kill them, and set the patient free of cancer. And hormonal therapy, it's basically works with tumors where the hormones play a role, like breast cancer or prostate cancer or uterine cancer or ovarian cancer. We can use the hormones to limit the growth of cancer, basically. So this is the idea or the lines of cancer treatment. Of course, you guys have seen the chemotherapy infusion stations in the offices you visit. Most of you have seen the radiotherapy. Surgery, of course, we have a lot of surgical centers that do the biopsies for the cancer. And you guys might came through the targeted therapy by next generation sequencing, or get questions about the next generation sequencing. And immunotherapy, I know Lauren has come through, the lady who the sales rep who sells the Solaris drug, and the hormonal therapy. So this is basically the idea. We didn't have a lot of time this episode to go through the hematology part. Maybe we're gonna postpone this for the next week. I hope you guys made benefit of this week's episode. If you have any questions or comments or anything, just go ahead and share with us. I'm sorry for getting you guys late, but I hope you guys made a lot of benefit out of this. Thank you. Go ahead if you have any questions, or say goodbye.
Thank you, Karim. I'm not sure. I missed. Did I miss something at the beginning?
I don't think so, but don't worry. The episode is recorded. I'm gonna send you guys the YouTube link, so you can always check the episode again. Okay.
Okay. And what about the test? Did you send us test already, or you will do it, or?
No, it was the same test from the last time. So you guys already have done the test, so don't worry about the test this time. You've already done this. Again, for the next time, we're not gonna have a test because we're gonna go go to the hematology part. It's the same PDF for the handout, the reading, and the same test. So don't worry about the test for not this time, not the next time as well.
Okay. Also, the thing is, we have, we validated next generation sequencing, right? Just for solid tumors. I think at least in New Jersey, right? But we
Not
cannot.
for all solid tumors, just few tests like the KRAS and NRAS and EGFR, but not for all. Of course, not for all solid tumors. Not all tests. There are like a couple hundreds of them.
Okay. And for hemangs, we don't have anything for now.
For what?
For hematologist oncologist, for this kind of cancer, we don't have anything. We are calling on hematologist oncologist mostly, right?
Yeah,
And
we are still working, so we're gonna develop more of the next generation sequencing, and we're gonna develop marketing material for them as well. And we're gonna provide like a specific detailed training about next generation sequencing, so that when we start calling on the hematologist oncologist for next generation sequencing, we know what exactly we sell, because most of the doctors, they don't have a lot of information about it yet, so we have to educate them about the next generation sequencing. It's a pretty hard goal to achieve, but now we are going through the basics. Maybe in later on the time, we're gonna have more knowledge that enable us to go through the specific details of next generation sequencing.
Okay.
Okay.
Thank you.
Sure, you're welcome. Thank you, guys.
Thank you, Karim. See you next Wednesday.
See you next Wednesday. Okay. Bye bye, guys.
Bye. Thank you.
Lauren, you have a question?
I know, but good job. Thank you so much.
You're welcome. Thank you. Okay. Anybody else?
مع السلامة يا كريم.
الله يسلمك. ألف شكر. جزاكم الله خير. ألف شكر. مع السلامة.